Welcome to the Peak Studio 12 DIA video tutorial. In this video, I will be going over the DIA workflows available in Peaks 12, how to set up the identification and quantification workflows, and how to view and export the results. You can use the chapters along the progress bar down below to jump to the sections that are relevant to you. The Peaks 12 DIA solution offers 3 different workflows: identification, quantification, and DeepNovo-peptidome. This video will focus on identification and quantification. The DeepNovo-peptidome workflow will be discussed in another video. The identification workflow has 3 different steps: a spectral library search, a database search, and de novo sequencing. Each of these steps are optional, so you can choose to run all 3, choose 2, or run only 1. This allows for greater flexibility, particularly if users wish to run a library-free search. There is also the option to run a quality control step at the end to assess quality of your replicate samples. This stepwise approach allows the search space to remain well controlled, leading to improved proteome coverage and increased accuracy. The quantification workflow involves the spectral library search, the database search, a label-free quantification step, and the quality control step. Like with the identification workflow, you can choose to run both spectral library and database searches, or only one of the 2. Now, let's jump into Peak Studio 12 to take a closer look at the workflow setup. Once you have opened Peaks 12, click on the New Project button to start a project. Here you can name your project and choose a project location. Click on Add to add your raw sample files. Make sure the acquisition type is set to DIA. You can then add your samples. This button will create a new sample for each file. Now, if you want to change any of the parameters, you could simply change the desired parameter, highlight the sample, and click Copy to Whole Project, and it will change the parameter on all the samples instead of having to do them one at a time. Now you can click on workflow selection and go straight on to the workflow setup. However, we usually recommend that you click finish at this step and allow your samples to load in. This will save a lot of time during the analysis. Once your samples have loaded in, click on New Analysis to return to the analysis wizard. You'll notice that your samples now appear under this In Project section of data selection. You can now highlight all of your samples and use this button to create a new sample for each file. You'll notice that you can no longer change the parameters because they've already been loaded in. Now we can proceed to workflow selection. Now we choose the workflow that we'd like to perform. We're going to choose identification for this part of the tutorial. Next, we can choose to set an error tolerance or allow Peaks to auto-detect the ideal error tolerance. This is checked by default. If you uncheck it, the default parameter mass and fragment ion tolerances will be set automatically based on your instrument type that you selected. You can change this, but we do recommend initially leaving the default values and only adjusting if necessary. We're going to leave auto-detect on for this example. Peaks 12 has a new option to select match between runs at the identification step. This feature is best used when you're expecting similar identifications across all samples, such as when you have replicates. Next comes the library search parameters. Check or uncheck the library search parameters box to choose whether or not to perform the spectral library search. Click on New to configure a new spectral library. Browse to find your compatible spectral library. I'm going to be using a .tsv file exported from Peaks 12. And give your library a name. Click on Add Update. The search can be filtered by peptide length, precursor M over Z, or charge. Leaving all these boxes unchecked will mean that no filter is applied. There is also the option to select a database for protein inference. This is a great option if you are only performing a spectral library search and is required if you are only performing a spectral library search plus a label-free quantification with no database search. Now we can select the database search parameters. Check the database search parameters box to run the database search step in peaks. You can leave the enzyme as specified by each sample because we already set the enzyme in the project creation step. There are two options for digest mode if your sample was enzymatically digested: semi-specific and specific. Specific will only allow digestion strictly following the digest rules of your chosen enzyme. This is a great option to keep a well-controlled search space. Semi-specific allows for one end of the peptide termini to disobey the digestion rules. This option opens up the search a little bit more and usually results in greater coverage. You can also choose the number of missed cleavages to allow. Click on Set PTM to choose your fixed and variable PTMs. For DIA, we usually recommend limiting the search to 1 or 2 variable PTMs to keep the search space controlled. You can also set the maximum allowed variable PTMs per peptide. Next, you choose your target database. Click on New to configure a new database. You can choose the FASTA format. The options are UniProt, NCBI, and Other. The parsing rules can also be customized under Advanced Options. Give your database a name and browse to find your FASTA file. Click Open. You can choose to validate your FASTA file to ensure that there are no issues with the format. Click Add Update. Be sure that the number of sequences that pops up in this corner here matches up with what you expect for your database. If it shows 0 sequences, it means that something has gone wrong. You can also choose to set a contaminant database. The two databases will be concatenated and searched together in a single round search. The database search can be filtered based on peptide length, precursor M over Z, fragment M over Z, or charge. Finally, you can set the parameters for the de novo search. You choose your PTMs in the same way that you do in the database search. Checking the QC step will give you statistics about your data attributes and identification attributes across your samples. You can pick and choose which attributes to include in the report. The report page is the last step of the workflow setup. You have the choice between sample-based and project-based filtering. If you choose sample-based, you set a -10 log p-value, which each precursor will need to pass to be included in the report. If you choose project-based, you set a false discovery rate at either the precursor or the peptide level. You can also either set the protein -10 log p or the protein FDR. You can choose to save the workflow parameters, name your analysis, and decide whether or not to export the results. Choosing to export will generate all possible peaks reports and can greatly slow down analysis times. We usually recommend users to select their desired exports after the analysis is complete. Click on Finish to start the analysis. Now let's take a look at some example results. When your analysis is finished, you'll be able to click on it in this window here. Click the drop-down menu to see the results from each step of your analysis. Let's take a look at the spectral library result first. You'll first be taken to the summary page. At the top of the summary page, you'll have the option to adjust the protein filters. The rest of the page contains various statistics about your results. You have a false discovery rate curve, retention time calibration curve, median delta RT across all MS runs, a table with your statistics of your data, result filtration parameters, PTM profile, distribution of precursor mass error, and a table showing the number of identified peptides by number of missed cleavages. Now let's click on the Proteins tab. Here you see a table view showing all of the identified proteins in this analysis. Click on Protein View Filter to filter which proteins show up in the table. Click on Show Hide Columns to decide whether or not you want to show sample coverage, sample precursor counts, or sample areas in your chart. Using this box here, you can also search for a particular protein of interest. You can search for accession, description, or gene. You can also export the proteins table directly from this page. In this window below is the coverage view. Click on a particular protein in the table to display the coverage view for that protein. You can switch between a full coverage view showing each of the supporting peptides in blue, or you can choose an outline view showing only overall which sequences have been identified. You can also change how many amino acids are displayed per line, whether or not there's a gap after every 10 amino acids, and whether to display the sequence based on FASTA or tryptic digest. You can also decide whether or not to show confident modification sites or show particular PTMs. You can switch to a full screen view of the coverage tab. And you can choose to copy the protein sequence, save an image of the protein coverage view, or get some coverage statistics. Clicking on this Peptides tab here will show a list version of the supporting peptides. Now let's click on the Peptide tab. Like with the Proteins tab, you also get a chart, this time showing all of the peptides that were identified in the sample. Only unique peptides are displayed here. If a peptide was identified more than once, only the one with the highest -10 log p-value is going to display in the chart. However, the sample areas are a sum of all of the areas associated with that peptide. Down here is the feature selection breadcrumb. We click here where it says sample 5. We can see each sample and whether or not they contain a feature for this peptide. By default, the one with the highest -10 log p-value is displayed. Click on a different feature to display the MS2 spectrum and precursor views for that feature. Clicking on a particular peptide, you'll be able to see in this window down here the corresponding MS2 spectrum. By default, a mirror plot is shown, showing the query spectrum against the library spectrum. You can switch this view to an annotated spectrum view. Hover over the peptide to show the supporting ions. Click these arrows to display all peptide spectrum matches associated with the precursor. Click on Spectrum Annotation Settings to decide which ions and which information you want to be displayed on your MS2 spectrum. You can switch between absolute intensity and relative intensity for the y-axis. And you can use these buttons here to zoom in on the X and Y axis and use this button here to zoom back out to the default view. You can also choose whether or not to show the alignment or the error map. You can also zoom in on the X or Y axis using your mouse wheel. Clicking on this button here will show the protein that this peptide is associated with. The ion match table displays the supporting ions in a list view. Ions that are displayed in color mean that they were found in your MS2 spectrum. To the right here shows different precursor views. Here is an overhead view of the retention time versus M over Z, with the feature being outlined. Hover your mouse over this little dot here to show the M over Z value, retention time mean, charge, and area. You can zoom in or out of this chart and use this button to center it again. You can also switch between a 2-dimensional and a 3-dimensional view. You can also export the LCMS view. Over here, you can see a precursor profile view, as well as a fragment ion extracted ion chromatogram view. Now that we've taken a look at the spectral library results, let's take a look at the database search results. The DIA database search summary view is going to look exactly the same as it does in the spectral library search results, only this time it will contain statistics about the database search. Let's look at the Proteins tab. You'll see that this, again, is the same as it displays in the spectral library search result, except now it includes proteins that were identified through the database search. The information in the Peptides tab for the database search result, again, is very similar to the information you see in the spectral library search result. The major difference is in the MS2 spectrum. Instead of displaying the query versus library spectrum, this shows the query versus the spectrum that was predicted by the deep learning algorithm. Finally, let's look at the de novo result. This is the DIA de novo result summary view. At the top of the page, you can adjust the de novo ALC score. This will change what is displayed in the de novo tab. On this page, you get your statistics of data, as well as some information on your search parameters. Let's look at the de novo result tab. Much like in the library search and the database view, we see the peptides found with this search in a table format. Contrary to the database search, instead of a -10 log P score on the peptides, we see an average local confidence score for each peptide. Hover over the peptide in the table to see the individual local confidence for each amino acid. By default, the peptides are sorted from highest to lowest average local confidence. Click on the table header to resort based on that category. Now we're sorting from lowest to highest ALC. If we hover over this peptide, we can see which amino acids are the low confidence ones. If you wish to filter out low confidence amino acids, you can use this button here. You can choose to show a mass tag for a confidence less than the desired amount. Let's say we move this to 80%. Now, any amino acid with a confidence less than 80% shows up as a mass tag instead of the amino acid. You can choose to export the table with these filters applied. Use this drop-down menu here to switch between samples. Like with the other results, you can switch between a mirror plot and an annotated spectrum view. Additionally, you can use this button to show all possible de novo candidates. And those are all the results for the DIA identification workflow. Now let's take a look at a label-free quantification analysis. Now I'm back in the analysis wizard at the workflow selection step. Let's select the quantification workflow. The Spectral library setup and the database search setup are going to be exactly the same as they were in the identification workflow. Now here's the label-free quantification step. New in Peaks 12, users can choose whether to prioritize high precision or high accuracy in the label-free quantification. High accuracy imposes a stricter error tolerance on the peptide fragments chosen for quantification. You can expect to see less peptides quantified with this mode, but a more accurate result. Whereas in high precision mode, you can expect more peptides quantified, but less accuracy in the result. In a label-free quantification, you'll need to group the samples into at least 2 groups. Highlight the samples you wish to include in a group and press this button to create the group. You can change the group name and the color associated with the group. Now let's create a second group. For DIA label-free quantification, it is required that you use all of the samples in the analysis in the label-free quantification. You can select the retention time range that you want to include, as well as the base sample. You can also choose average instead of a single base sample. Click on Peptide Feature Filters. You can filter which peptides are used in the quantification based on their average area, their quality, or their charge. The quality scores how quantifiable a feature is. Factors that affect the quality score include the similarity in M/Z, retention time, extracted ion chromatogram shape, isotope distribution shape, signal-to-noise ratios, frequencies, and intensities between the different features. You can also decide how many confidence samples you need to have per group in order for a peptide to be included in the quantification. Setting this to 0 means that the peptide does not need to be present in every single sample in order to be included in the quantification. If you have samples which you know contain certain peptides in some samples and not others, but you still want to include those peptides in the quantification, you should leave this at 0. Check Use in-Group Coefficient of Variation filter to require at least one group to have a coefficient of variation less than the CV threshold. Clicking on Protein will give you some options for protein filters. You can choose to set a significance value or use an FDR. The significance is a -10 log p-value, where the p-value is derived from the significance testing method. You can either set this significance value yourself or choose an FDR, in which case the significance level will be chosen based on what is required to meet your FDR. You can also filter based on fold change. Both the significance and the fold change can be changed pretty easily in the summary view, so we typically recommend that users just leave these as they are. You can also choose the minimum number of peptides required in order for a protein to be included in the quantification. If you check modified form exclusion, and a peptide has both modified and unmodified forms, only the unmodified form will be used in the protein ratio calculation. Finally, you can choose your normalization method. Choosing no normalization will mean that no normalization is performed on the sample. We usually recommend users to start here if they're unsure of what method to choose. If you check Use TIC, Then Peak Studio will use the total ion current of the samples to calculate the normalization factors. This option is best used if you expect a consistent protein expression level across all of the different samples. Manual input will use the same calculation as TIC, except you'll be able to set your expected ratios. So for example, if you have one sample that you know is double the concentration of another, you can set a 2-to-1 ratio. You can also choose to use internal standard proteins. This will only become an option if you modify a previous analysis where you have a library or database search result. But you can choose particular proteins that are consistent across all of the samples to normalize to. With the label-free quantification, the QC step will have some more attributes that you get to choose from that are based off of the label-free quantification. And your report options are going to be the same as they would look in the identification workflow. We can now click Finish. Now let's take a look at a sample label-free quantification result. At the top of the summary page, we can change the protein significance threshold or change it to an FDR. We can also change the fold change. This will change what is displayed in the protein profile heat map, the volcano plot, and what we find in the Proteins tab. There are also some additional figures available. You can get sample correlation at the protein level, the peptide level, and the feature level. You can also get the group correlation, again at the protein, peptide, or feature level. There's also a density ratio plot. And an interactive heat map. Click on a protein or protein cluster to see a variation trend chart. You can choose to export this trend chart as well. As mentioned, the summary page includes a heat map, a volcano plot. You also get a retention time and M/Z shift distribution, PCA plot, protein covariance values, result filtration parameters and statistics, and a summary of your search parameters. The Proteins tab will look fairly similar to that in the Spectral library and database search result. Except now you get individual heat maps per protein at the sample level and the group level. Hovering over these heat maps will show the individual areas and calculated ratios. Like with the identification results, you can either look at a coverage view or a peptide list view. In the Peptides tab, you also get the individual heat maps per sample and per group at the peptide level. You can also see some feature details below. By default, one sample per group is shown, but you can check or uncheck any sample you wish. The precursor profile, as well as the fragment ion EXI-C profile, are also shown. And that is an overview of the label-free quantification result for DIA. Let's take a look now at exporting your data. Click on the Export tab under your analysis. Here you will be able to see all possible exports. They will be sorted by the steps in your analysis. For the DIA result, we see a spectral library, a database search, and a label-free quantification. Some of the possible exports include CSV files with your identification and quantification information, FASTA files, and HTML exports. To export a spectral library for use in further DIA analysis, you can select Spectral Library TSV under Database Search Exports. This option will also be available on a de novo result. Once you have selected all the results you would like to export, click on the Export button. It will be saved into the path here, which you can change. You will get a pop-up when your exporting is complete. We hope you enjoyed this tutorial on the Peaks DIA solution. For more information, you can visit our website at bioinfor.com.